This is Key Assessment 6A, which is the Component 2 stuff that we're doing. We've got a photo micrograph, so it's down a light microscope showing root tip squash essentially. So this is all about cell division. Name the type of cell division taking place in this tissue. Well, there's two types, mitosis and meiosis. How many new cells are being formed? That's the key to guessing which one it is. Are these cells dividing into two, or are they going into fours? And I can just see things going into two there, so that's going to be our normal mitosis. Count the number of cells in metaphase and anaphase. Right. So metaphase, I always remember it, meta, M, middle. So where the chromosomes are lined up at the equator. So that appears to be one. Yep. That's another one. And metaphase, there we are. So I reckon there's 3 in metaphase. And anaphase. So anaphase is just like this one here, where the chromatids are attached to the spindle and being pulled towards the equator, but we've not yet disappearing, not yet forming the nuclear envelope, so we can still see the chromosomes. So I reckon that's one. That's one. There we go. These are anaphase. That's anaphase. That's anaphase. Maybe not. I'd be into disappear there, but that one possibly, yeah. So 1, 2, 3, 4, 5, 6, 7. So 7 in anaphase. Calculate the time taken for the two phases using this formula. So the time there, which is going to go in here, is number of cells in the phase divided by the total number. So for metaphase is number of cells 3 divided by total number of cells. Totals of the cells 64. So I'm sure if we counted all of those, there'd be 64. 3 divided by 64 times by total time. There's the time, and there's a total 720. So 3 divided by 64 times by 720 is 33.75. So they've given it to the nearest minute, so I'm going to give that to 34. So 33.75. But I'm going to go with their level of precision that they've used. Anaphase, I'm going to do the same thing. Anaphase, number of cells, 7. Divided by total is the same, 64. And the total time is the same, 720. So 7 divided by 64 times by 720, and we got 78.75. So I'm going to go for 79 there. Oh, I could check these add up, but I'm not going to do that. Calculate the percentage time spent in each phase. So a percentage is just our value divided by the total times by 100. And so our 34 divided by 720 times by 100 is 4.7. So percentage is the value divided by the total times by 100. So in metaphase that was 34 divided by 720 times by 100 equals 4.7. And again 4.7, I'm going to round that up to 5 there. For anaphase, that is 79 divided by 720 times by 100. So 79 divided by 720 times by 100 is 10.97. So I'm going to round that up to 11. The examiners will carry these marks forward if you made an earlier error, or there is an acceptable range of answer there. Describe 2 ways in which the process of cell division shown differs between plant cells and animal cells. So this is just AO1. Can you remember the difference between animals and plant cells in terms of mitosis? So animal cells, there are centrioles. That's just one of the things that you remember about mitosis from when you've done that topic. And of course there's no cell plate in animals, forming that kind of cell wall eventually. So in animal cells, again, there is no cell plate forming. There'll just be a constriction of the cytoplasm. So 2 marks there. And over the page, abnormal growths called tumours, and the term applied to the formation of malignant growth. So essentially the name of that disease, which is cancer. And studies have shown that the plasma membrane cells have some transmembrane proteins. So trans means across the membrane proteins with shorter carbohydrate side chains. Okay, draw diagrams of a short section of the plasma membrane from a normal and abnormal cell. What do we need? We need the following. We need phospholipid bilayer. We need the transmembrane protein and these carbohydrate side chains. And these are attached to the protein. So we just remember our normal membrane structure. So there's our phospholipid bilayer, transmembrane protein. So this is my transmembrane protein, and carbohydrate side chains. So something like that. And then we've got more phospholipid bilayer. Include labels for the following structure, so we better include our labels. So this is our phospholipid bilayer. We need a transmembrane protein. So this is my transmembrane protein and carbohydrate side chains. So carbohydrate side chain. So we've done everything it said in the question. And then the abnormal, that's going to be shorter. So I'll tell you what, we could make these long. and we could do essentially the same diagram, same transmembrane protein with same but we're going to have shorter carbohydrate side chain. So shorter carbohydrate side chain. I don't know whether you'd have to label everything again, but I think we labelled it once, so that's enough. The marks are phospholipid bilayer, transmembrane protein, carbohydrate chain labelled, reasonable quality drawing showing that phospholipid bilayer and the transmembrane protein with carbohydrate side chain on, and the second drawing having a shorter one. If you did this in the tower room, it was easy because there was a big poster up showing exactly what you needed. How would the difference described in 2 affect the properties of the abnormal cells? Right, well, you might not know much about cancer at all, but just go with what you do know. So this is things like remembering that these carbohydrate side chains are to do with cell recognition, cell signaling, communication, that sort of thing. So the abnormal cells not able to, for example, in cell recognition or signaling or communication, something like that. And the other thing I'd also say is the receptors might not work. So if you think of signaling, like hormones and things like that might bind to these side chains and enable the cells to be recognized, that sort of thing. So we want two separate points being made, and recognition, receptors are the kind of the things that you know about already. And I'd also maybe put that word binding in somewhere, so e.g. Will not bind. So that's what you know, but you're just applying it to this new situation.